we continue our discussion on DNA replication, and in this video we will get a glimpse of origins of replications. And specifically, we will explore them in the context of bacteria, Archaebacteria, and eukaryotes. And that'll be mainly yeast and mammals. Let's start by highlighting the motivation to study the origins of replication. This is actually fairly straightforward. DNA as a storage molecule is double-stranded, and if you want to replicate the double-stranded molecule, you will have to unwind it into single strands. And the origin of replication, as we will see, helps in this process. To get an intuition about the origin of replication, let's actually start with the tractable case of eukaryotes, because they have a linear chromosome. And for simplicity, consider a chromatid. This is the structure that you have probably seen in textbooks. And this has two open ends, which are called telomeres. And by open ends, I mean that they have overhangs that can be used directly for potential unwinding. This may in simplicity look something like this, but in reality, the telomeres have a T-loop cap, which is just a folded structure. But the point is that there is a 5' end available if you were to unzip the DNA at the telomeres. And you can do this same process on both sides. By the way, if you haven't noticed a technical mistake, I started out by suggesting chromatid for simplicity. But chromatid is a condensed structure found in mitosis. DNA replication occurs in the interphase, and there is no chromatid in the interphase. If you actually look at the DNA in the interphase, it is present as a somewhat organized hairball. In any case, the point remains that you have two ends on each chromosome that can be used to prime and start DNA replication. And this replication then has an inward direction. But if we take this intuition, we run into the problem of different chromosomes and different timings of replication for each chromosome. Also, different chromosomes have different lengths. So the speed of replication has to be calibrated for each chromosome. You see, this gets messy, so you cannot just start from the ends. At least it doesn't work intuitively. Eukaryotes have linear chromosome, but if you look at prokaryotes, they have circular chromosome. So there is no way to start replication from an available end, because there are no free end or an overhang in circular chromosomes. This means that the replication must start internally, which means that the chromosomes, or DNA specifically, must have a way to open internally to allow the replication to proceed in some direction. And it is this internal site, or a DNA sequence specifically, that is called the origin of replication. In an organism, you need at least one origin of replication. But in some complex ones, as we will see later, you can have thousands of origins of replication. The postulation of origin of replication came from a model called Replicon Model, back in 1963 through studies in bacteria by Sidney Brenner, Francois Jacob, and some other prominent scientists. It suggested that in the bacterial chromosome there is a site that allows for the chromosome to be replicated, and that contains the origin of replication. Now, note that the bacteria has a single chromosome. A Replicon model suggested that each bacterial chromosome has one origin, or ORI for short. And this chromosome then can also be called a single replicon, because it replicates itself. So a bacterial chromosome is also a replicon. Now contrast this with eukaryotes, where the chromosomes are very large and linear. They have multiple origins for each chromosome, which can independently start replication, which means this part is one replicon. Then this other part is another replicon, and so on. So in eukaryotes, one chromosome can have multiple replicons in it. Okay, let's get back to the Replicon model. The Replicon model suggested that there is a replicator sequence and an initiator factor, and both are required for the initiation of replication. The replicator is a DNA sequence, whereas the initiator is a protein factor. Before we dive into the details and complexity of the origins, let's get a general idea about the replicator and initiator. We will start with the replicator. As we just said, replicators are DNA sequences, but they are sequences that aid in the recognition of the origin. So consider this piece of DNA, which can attract some proteins and license that region as an origin. Then, this piece of DNA is called a replicator. Within the replicator, you start to unwind the double strands into single strands. And that particular location of unwinding is called the origin of replication, which is the starting point for replication. So the origin, or ORI, is part of the replicator sequence. Analogically, this is similar to the situation in transcription, where you have the promoter and a TSS. The promoter is the recognition sequence for starting transcription, but the actual start point of the transcription is at the TSS. The other part of the replicon is the initiator. This can be one or more proteins that recognize the replicator. Once again, the replicator is the piece of DNA that aids in the recognition, and the recognition is done by these proteins that bind to it. So initiators have motif binding potential. They recognize the replicators. And once they do that, the initiator can recruit other proteins like helicases and polymerases and initiate replication. A very subtle but important point is that DNA polymerase and helicases do not actually recognize any specific DNA sequence motif. In this respect, these initiators are very important since they are the only proteins to have some DNA specificity. To make this point very clear, let's again compare this with the transcription, where you have the promoter and the TSS. The transcription factors recognize the promoter sequences, and they in turn recruit RNA polymerase onto the promoter. The RNA polymerase by itself does not recognize the promoter. One last thing before we deep dive into origin of replication, I want to stress that the Replicon model was meant mostly for bacteria. The origins in eukaryotes, as we will see later, are slightly more complicated and do not really fit the constraints of a Replicon model. Now we're ready to take a deep dive into the bacterial replicator and initiator. We will start with the replicator first. Bacteria have a single replicator, as we just said. They have one replicon, one origin, and one chromosome. The replicator is called origin, OriC for short. This stands for chromosomal replication origin. The replicators across different bacteria can be very diverse in length. They can go anywhere from 250 base pairs to as big as 2,000 bases. But in common, they're all recognized by the same DnaA protein. As we will see shortly, the DnaA protein is the initiator protein. Back to the replicators for now. They can be either continuous or bipartite. Let's understand the structure of a replicator by taking the example of E. coli, which has the replicator, which is around 250 nucleotides in length, so a fairly compact replicator. This replicator has some signature components. First, it has 3 sequence repeats, which are 13 base pairs in length. They are sometimes just called left, middle, and right motifs. And together, they make up the DU element, which stands for DNA unbinding element. Then you have 4 more important motif repeats, which are 9 base pairs in length. They are simply named as region 1, region 2, region 3, and region 4. And they have a specific directionality to them. This entire region is called DAR, which stands for DnaA assembly region. These 4 regions are high-affinity sites for DnaA protein. Actually, they're always bound by DnaA proteins. Within this DAR region, you also have some other small sequences which are also about 5 to 9 nucleotides in length that look like R1, R2, and R3 and R4. The names of these internal sites are not that important, they're just named R5, Tau, I, and C sites. These internal sites are low affinity binding sites for DnaA protein. The binding of DnaA to these other sites is ATP dependent. As we will see shortly, DnaA has an ATPase subunit. There are also some other sites within the DAR region that affect and regulate the activity of DnaA proteins, and these are IHF and FIS sites. We'll discuss the function of IHF and FIS sequences when we talk about the mechanism of DNA unwinding in a different video. The last part of replicator is this newly discovered region between DAR and DU, which is named as DnaA trios, which contains multiple GAT motifs. The function of this region is to aid in DnaA oligomerization into the DUE region, and this helps in the unbinding of the DUE region. Since DUE is unbound, we can say that the DUE is the actual origin of replication. So in summary, the DnaA protein binds to DAR regions and unbinding occurs at the LMR DUE regions. The DUE region, after it is unbound, is also the site for single-strand protein bindings. Details of this will come later when we discuss the exact mechanism. For the sake of replicator, you just have to take note of DUE, trios, and DAR region. One side note, I mentioned this bipartite origin in the beginning. So let me make this clear. In other bacteria, the general structure of the replicator is very similar in a broad sense, that you have a DUE region and a DAR region. The length of these DARs and DUEs are variable, but the syntax is pretty much the same. In some bacteria, for instance the Helicobacter pylori, the origin is split into two parts, so it is a bipartite structure. One part of the origin is a DAR region exclusively, and then at some distance there is another part which contains another DAR and the DUE region. So it has this split structure of origin, and that is why it's called a bipartite structure. Alright, that was the replicator. Now we get into the initiator of this replicon. As we said already, the DnaA protein is the initiator in bacteria. It is common to all known bacteria. The structure of DnaA is fairly simple. It has 4 domains. 1 is an oligomerization domain, 2 is a flexible linker, 3 is the ATPase domain, which is of the DnaA+ family. The AAA stands for ATPase associated with various cellular activities. And this is where the AAA comes from. The plus symbol is a nomenclature to differentiate it from the non-plus AAA family. This domain also binds the single-stranded DNA. And DUE gets unbound, so that's one binding target for this particular domain. The third domain also recognizes in some capacity the DnaA trios region. The main recognition of DnaA, however, of the replicator comes from this fourth domain, which contains a helix-turn-helix region. This one is very important. It functions to bind the double-stranded region. More specifically, it binds the DAR region 1, 2, 3, and 4. And this snapshot completes our discussion of the bacterial origin of replication. Let's now talk about Archaebacteria. They typically have 1 to 4 replicators per chromosome. Their recognition sequence are called origin recognition boxes, or ORBs for short. And they're very diverse in composition. As an example, if this is an Archaea chromosome, then it can have anywhere from 1 to 4 origins that may or may not be different from one another. If we zoom into one of these origins, you will find a DUE element and a bunch of recognition motifs called ORBs. As I said, these ORBs can be very different from each other. That is what the colors represent here. So that's the general structure of the replicator. The initiator in Archaea is called ORC1 or CDC6. This protein binds ORBs. In Archaebacteria, there can be multiple paralogs of ORC1, conveniently named as 1, 2, 3, and so on. These paralogs are different in their affinities to different ORBs. ORC1 looks very similar to a DnaA protein, in that it has a AAA+ domain and a winged helix domain, and this serves as the DNA binding region. Now, let's put this in context to the replicator. The different origin recognition complexes in one or more combination can recognize specific ORBs. So a replicator can have multiple paralogs bound to it. This together unbinds the region, which is the DUE region, and that can recruit helicases, polymerases, and then start the DNA replication. Alright, that is the context of Archaebacteria. Now let's move on to the eukaryotic replication origin. Eukaryotes have many origins. Broadly speaking, eukaryotic replicators or origins can be split into yeast and everything non-yeast. Let's go over yeast first. They have a 12 megabase long genome, and when I say yeast, it encompasses all the known Saccharomycotina species like Cerevisiae, Pombe, and others. yeast is the only known eukaryote with a specific consensus DNA sequence for origin of replication. And the replicator in the yeast is called ARS, which is short for autonomously replicating sequence. There are about 300 to 400 of these in the entire yeast genome. Let's sketch it out. It has 4 important motifs. The A motif, which is about 11 bases long, and the other motifs are variants of the B motif named B1, B2, and B3. The A and the B1 is the core sequence, which recruits ORC. The A motif has a conserved sequence which is fairly AT-rich, and this entire region is around 100 to 200 bases long. And because now that we're in eukaryotes, you have to note that the DNA is maintained as a chromatin. So you have histones that may be important to the origin recognition. Functionally speaking, the core A and B1 motifs recruit the initiator ORC. The B2 motif potentially recruits helicase. The function of B3 is somewhat unclear except from the fact that it recruits this ABF1 factor, but it doesn't seem to be essential. So it is not clear why or how this is important. The initiator in the case of yeast is this ORC, which stands for Origin Recognition Complex. It seems to be conserved in all eukaryotes. It is a complex of about 6 proteins, which together form a ring-shaped complex. Just to intuitively give you an idea, I will sketch this out. The ARS recruits the ORC at the A or B1 motif. Specifically, the winged helix domain recognizes the core. The ORC also has AAA plus domains, so most of the binding is done at these A and B1 motifs. The ORC can also interact with some specific histone marks, but they may not be functionally as important. Once this recognition is complete, the origin is opened, helicase is loaded, and the replication can start. So that was yeast. Now let's consider the rest of eukaryotes, and I will limit the scope and generalize them by considering mammals. Outside of yeast, the eukaryotic origin of replication is actually quite puzzling. It is still an active area of research, because there is no clear consensus on the replicator sequence. People haven't found it, or maybe it doesn't exist. But it is clear that origins are present at some specific locations in the genome. Usually they are in the order of thousands, but only 10 to 20% of them are actually used. This phenomena is called origin licensing. The origin definition in eukaryotes depends on multiple factors. The lack of consensus is thought to be overcome by some other variables, such as DNA accessibility, AT or GC-rich regions, CPG islands, nucleosome positioning, epigenetic marks, and even DNA topology, which is typically G4 motifs, also known as G quadruplexes. These multiple factors make up the prevalent view of the origin of replication in eukaryotes. Let's see what this means. Places of origin of replication means that there are regions which in some capacity contain these elements and somehow recruit ORCs at these locations. The ORC being the initiator protein complex. These ORCs can interact with histone marks such as H4K20 methylation. Elements nearby can also recruit some other specific proteins, and the synergy of all these elements can lead to helicase recruitment, and then opening of the DNA can happen, and this can initiate the replication. And this region where all of these occur is called the initiation zone, which is typically around 30 to 40 kilobases in length. There is another view that has recently gained precedence, which mainly relies on 3D genome topology. And specifically, it is the topologically associating domains which are in context. These dynamic structures, TADs, are regions of DNA that are much more frequently interacting with each other. The edges or borders of TADs are marked by CTCF proteins, and the dynamic organization is mediated by a motor protein called cohesin. The point here is that borders, which are often 40 to 50 kb in length, are highly enriched for CTCF protein binding. The movement of cohesin and concentration of CTCF in these locations is thought to create a permissive environment for helicase or ORC loading at these borders. The mechanism at play is called loop extrusion, which is the name of the movement done by cohesin. Exactly how the loading happens is unclear. This may sound all wishy-washy and directionless, But if it does, that is exactly the point. As I said in the beginning, this is an active area of research. Anyways, this completes our discussion on the origin of replication.